All right, so as the mics are getting passed around, I can kind of open this session up and maybe start with Gültekin on your talk on the pipes and viz working group. You mentioned something that Jamie and I also saw in the working group, in that it's called pipes and visualizations, but we didn't really see a need to be developing pipelines because there's really so many different pipelines out there. The major challenge in the community is having resources to guide selection and implementation of different pipelines. But what you mentioned that you guys are walking into is a huge need in the space of maintaining pipelines. So what sort of role do you see the working group playing in terms of maintenance of pipelines and workflows for pathogen genomics?
First of all, thank you, Kevin, for the question. I would like to thank you again, while you are next to me, about the legacy that you handed over to us. It was really nice to be a co-chair of the Bioinformatics Pipelines and Visualization Working Group so far. The initial idea was taking the legacy of you provided to us and try to take it a little bit more forward. So I know we had some validation data sets and pipelines that we created in the previous era, but in the near future, we will mainly focus on phage validated pipelines. These are all thoughts that we have, some of the projects that we are in mind. So what I mean by that, like, there are many pipelines out there, and you can go and work with it and have some results. But reliability of the results vary between pipelines to pipelines, and there is no standards. I mean, we have 10 simple rules for bioinformatics pipelines, but there's no simple pipeline, there's no simple results that you can get, and sometimes you may question the results that you're having. So in phage, we may come up with a solution to that, which can be phage validated pipelines. We can use the 10 best practices that we have, but we can create some benchmark data sets, and with the, of course, help of the community, we can validate these pipelines and make everybody sure that this pipeline is working very well, and of course it will be backed by some articles as well, of course. But yeah, I think in the near future, some of the projects that we are planning to do.
Very cool. Nice, great to hear. Just checking if there's any questions across the audience. I think we got some here, please.
Thank you. Hi, Khaled Youssef from NICD South Africa. My question to Emma with regards to the new platform, looking great. I have a question about the public health use, and would it be possible to consider trying to add a component of translation, the data that we generate and submit, and how can it be translated to public health eyes? And my understanding that most of the time we focus from academic perspective or scientific discoveries about new lineages. But when epidemiologists look at the data, whether it's lineage 1 or lineage 2, with the presence of these mutations, for them it's not complete. What is complete is to understand what are those lineages meaning from clinical surveillance perspective. And to my knowledge now, there is no database or no platform can put the guidelines for that. We actually doing it one by one in our meetings within our institutes. I come from Public Health Institute, and this is a main challenge that we have, because every time we have to sit around the table, discuss specifically during the SARS-CoV-2 pandemic, and also because I also work in wastewater space, and this is also another challenge for the wastewater findings. That's how do you translate it? How do you go beyond the idea of just doing the work and publishing the article? Because at the end of the day, you're looking for the impact.
So that's a great question, but I'm afraid you're not going to love my answer, because part of what's very interesting with the work that we do at Pathoplexus is as much as deciding what we are going to do, it's often also deciding what we aren't going to do. And one of the things that we at the moment, at least the executive board at the moment, has decided that we don't do is provide direct analysis or interpretation of the data. And this is really because we don't believe that we should be the ones that by default say what the analysis is or what the interpretation is. Science is done by many people, and who are we to say what the analysis is? Now, the other flip side of that is that even though we don't think that Pathoplexus is the one to say, this is the analysis, trust us, we know that that analysis, and as you say, even other resources, is incredibly valuable. So what we have with the API is a way to allow people to link from the data to tools. At the moment, we just have 2, but we're very young, and this isn't something that has to be developed by Pathoplexus. We can have any tools that maybe even people in this room, if you would like to see an mpox report format or a website that shows particular variants in another one of the pathogens we support, you can build that and we will link to it as long as you can accept that data coming in a very straightforward format. So for example, what this would mean is that you or maybe someone else could build a kind of mpox, for example, report format that explains in more friendly language what the lineages are circulating, what that means, what mutations they have, what the interpretation is, and you could link that out from Pathoplexus. What's nice about us not doing it is that also means if someone else wants a different report format or they disagree with what you're saying about the lineages, they can also make something that does that. So it means that we have a more democratic way of building the tools that we need, but then making sure they're available for everyone. So I hope that's a bit of a balanced answer, that the answer is, well, we're not going to do it, but we want to enable it.
Emma, as a follow-up to that question, though, with other databases, like, for example, GISAID, when you submit SARS-CoV-2 data, they do some kind of QC or original check. When they're looking at the data, so that kind of allows them to infer lineage, for example. And then that's actually the lineage that's included in GISAID. Is that not the Pathoplexus approach?
Yeah, so sorry, I should clarify. We don't do the more detailed analysis, but we do basic QC. So we do some quality control checks. We also do things like we list all the mutations between your sequence and the reference sequence. That includes amino acid translation, so amino acid mutations as well. And then also things like the lineage assignments and this kind of thing. And we're going to add more of these QC checks in the future to make it easier for people to, for example, exclude sequences below some certain threshold. So I guess that is analysis, you're right, but it's really just kind of things that help you to understand that sequence, not taking all the sequences together and doing something more in depth with it.
Fair. Please.
Yeah, thank you very much. I'm Samuel from MRC Gambia, but I'm speaking from experience because I work in Nigeria. My question, or maybe appeal, is to Emma. In your presentation, good one, but you mentioned the restriction period to be 1 year. And then I'm thinking, as someone that is coming from a low resource setting where priority is not given to publication. Now, I have the data, but I know that within that 1 year, I might actually get it published. So I will still have to keep it. So the question is this cast in gold, or you can just extend to like 2 years so that people that have, not everybody have the money to generate the data. You source for the money yourself from salary, student, paying for that data, and then you get the data. You still have to look for the money to do the publication. And then you are saying 1 year. Within that period, the salary is not enough to do that publication. So maybe you might have to consider that in settings too. Thank you.
This is something that we thought about a lot when we were starting Pathoplexus, and actually it's one of the things that we drew on the phage community to ask. How long should the kind of embargo period be? Should it be 3 months, 6 months, a year, 2 years, 5 years? And we talked about this a lot because we recognize that, to be fair, even if you submitted the publication the day after you got the sequences, with the review process and burdens that we have in journals today, you still might wait a year before you get that paper out. What we came up with this 1 year was essentially the consensus from everyone that we spoke to. At the moment, that is fairly set, as in that's in our rules, but it's possible that the EB and the members could decide to change this. But I guess I also want to put a little bit of emphasis on the other end, that I completely emphasize that it's a disadvantage for many countries that they cannot do analyses as quickly as some other countries, and that disadvantages people for publications. However, I think that the longer term answer is we need to really scale up how much we are funding this and how much we are expanding education and access. Because one of the biggest things, in my opinion, to enable data sharing is enabling data analysis. Because if you know that you're going to be able to get a paper, or at least a preprint, if you're worried about the review system, at least a preprint out quickly, If you know you can do that, you feel empowered to share. So it's not just about saying data sharing, data sharing, data sharing. It's about backing that up by making sure we have the education, the workshops, the training materials so people know how to do the analysis, and then funding, not just generating the data, but making use of it in the countries where it's being generated. And my hope, I know I'm being a little optimistic here, is that we start doing this better so that everyone can publish in a year.
Well said. Kathryn, can I ask you a question? You had the talk on the different plasmodium approaches. I think one slide that was kind of surprising is that there's a targeted amplicon approach, but there's competing panels. I think you had like 5 or 6 different panels. Can you explain what's going on there? Is it that so many people are interested in different genes, or are these largely overlapping with maybe just minor differences in primers?
I think this comes largely from the funding structure and what motivates projects, and a lot of the time the funding is, here's funding for one specific project, and then you build a panel, you build your pipeline, and then you analyze your data and put that data out. And the funding structure doesn't really motivate people to use the same panel. Although there are lots of groups where panels are being developed and there's multiple people using them, and that is becoming more common. It has just been the way that the panels have been expanding as people have jumped on kind of the bandwagon of making them. And now we do have an...
Please.
Hi, the question is to Racheal, and I was actually fascinated by your one of your drugs, novobiocin, and we actually picked it up when doing some in vitro work with TB. And what we were struggling to find is examples of how it is used in either human cell lines or perhaps even something more complex. And where we got stuck was that its primary use is in veterinary science. I was just curious if you had any kind of idea as to how its utility or its safety would work in a human cell line, for example.
Do you mind repeating that? Yeah. Do you mind repeating the last bit there?
Yeah. What I was curious with novobiocin, and you had it as one of your top candidates. You had novobiocin as one of your top candidates as part of the candidate screening, and you were trying to figure out how can you actually deal with antimicrobial activities. So the machine learning identified these metabolic biosynthetic enzymes.
Okay, I think you're talking about the pathways.
Correct, yes. The biosynthetic pathways.
So for now, we are still on the next stage of exploring what the different pathways do and how they actually contribute to the synergy of the drugs. Yes, for now, we have only done the machine learning, getting the drugs, and then also identifying the pathways from the genes that we actually used for the training and testing of the model.
Racheal, as a follow-up to that, with those combo therapies, is there an established benchmark literature of combo therapies that you could, validate what your machine learning models have proposed, or is this something that you got to go in the field and test?
So the machine learning model we benchmarked based on the pre-existing, some of the combination therapies that were either known to be synergistic or antagonistic. That is the test set. Our test set was basically drugs that are actually known to be synergistic and antagonistic, also the train set. But then for the graphs that I showed later, those are drugs that are, for now, we could call them novel combination therapies.
Got it. Thank you. We got time for one more question. Yes, please.
I would like to ask a question from Noshi. No? Noof.
Yes.
Okay, sorry. You were showing on one slide about the genotypic results for the antibiotics, and most of them, they were showing 100%. Okay, so did you check on the phenotypic side? Because we didn't see that slide where you were showing the phenotypic and the genotypic. So under the genotypic, yes, we can see it's 100%. But if you see on the phenotypic, maybe it wasn't even 100%. So did you do the phenotypic and the genotypic on the same samples that you were doing? Thank you.
Thank you. But in the beginning, I show you the AST phenotypic for those samples. All are resistant except one samples. Those for 55 samples. When we go through genotypic, we just did for 37 samples. And those the same, resistant. But we did by, I took first that I did phenotypic by 2 AST. One by MIC for colistin, and the other one for disk diffusion, for the routine antibiotics resistance. So all the samples resistance to those the same in genotypic. But we can see the genotypic Y 100%, then 49%, and so on. Those for just the 37 samples. But those 55 all done by phenotypic. And just for colistin, because I just took lastly, that this colistin we already published in another conference. We talk especially for colistin, because when you find some samples that resistance to colistin is not easy. So most of them should be susceptible. But this sample, yes, resistance to colistin just phenotypically. So we have to do phenotypic, then to check by genotypic.
All right, thank you all. Please give a round of applause to our panelists.